The process of proline hydroxylation, as I noted earlier, requires vitamin C. Without vitamin C, some really nasty things happen. A deficiency of vitamin C leads to the condition known as scurvy. Now, scurvy is a pretty serious disease. You can see in the picture in the lower right some drawings of a person who has scurvy. These are from ancient times, and the disease has actually been known since at least the 13th century. Crusaders that went off on long crusades in the 13th century went and took with them primarily meat and salted meat because they didn't have a way of preserving things. They didn't have fruits and so forth, and they didn't know of the importance of those fruits. But as a result of going off and eating a sole meat diet, they developed a condition known as scurvy because they got no vitamin C in their diet. They went away as big hulking brutes, and they came back literally falling apart. So the value of citrus fruits for preventing scurvy has been known for as long as 1497, when the explorer Vasco da Gama took off on a voyage. And he took with him citrus fruits because there was this understanding that perhaps it might make a difference. Well, despite that knowledge, and despite the fact that Vasco da Gama's sailors came back and they had no scurvy, unlike many other sailors at the time, over 2 million sailors died of scurvy between 1500 and 1800. Which tells you a lot about how slowly information actually gets out. Now, proline hydroxylation is important, as I noted earlier, for increasing collagen's thermal stability. Without it, it's not stable at body temperature. But that also means that you can break it down by heating things up a fair amount. One of the reasons we cook meat, for example, is to make the meat tender, and we're breaking down the collagen that's found inside of that meat. Now, collagen is stabilized at body temperature, as I said. The addition of hydroxyl groups to collagen raises the stability of collagen by about 15 degrees, a pretty critical thing. So I noted earlier the importance of lysine that appears in the collagen sequence. Lysine doesn't occur very frequently, but it's very important for making interstrand crosslinks or chemical reactions between individual lysines, as we shall see. These lysine-lysine bonds give collagen the strength that I've described earlier. Now, the reason that these happen is because lysines are made into an aldehyde form, as we shall see, and these aldehydes are readily oxidized. And as they oxidize, they react with each other and form a covalent bond. This, as I noted, improves the strength, in some cases as strong as steel. Lysyl hydroxylase, which is equivalent to the prolyl hydroxylase I described earlier, is the enzyme responsible for this reaction, and another enzyme known as lysyl oxidase can also catalyze it. Now, the reaction that you can see here on the very top of this slide is the reaction where hydroxyl groups are being placed onto lysine. Now, it doesn't have the detail of the prolyl hydroxylase reaction, but the process is actually very similar. We can see the hydroxyl group being added, and we can also see that vitamin C is needed, just like it was needed for the hydroxylation of proline. So again, vitamin C is necessary to provide those electrons to reduce the iron, just as it happened with the prolyl hydroxylase. Hydroxylysine found within the collagen is then secreted, and as I noted earlier, that hydroxylation is necessary for the secretion. In some cases, the hydroxyl group that's shown here is a target for glycosylation, meaning that sugar residues can be added to it that will help to modify and give the protein some of its characteristic features. The more common thing that happens, however, to the hydroxyl group is that it can be oxidized to form either an aldehyde or a ketone, depending upon whether it's as shown on the picture here or as whether it occurs on the end, as I will show you in just a second. The hydroxylation of lysine within collagen, as I described here, also has to occur inside the endoplasmic reticulum, because if that doesn't occur, again, the collagen will not be released. The lack of hydroxylation impairs secretion because, again, we're going to make weak collagen if those residues aren't present. So another reaction that can occur in the side chains of lysine residues within collagen is shown on the screen here. It also involves oxygen, and is catalyzed by the enzyme known as lysyl oxidase, as you can see here. This reaction, the amine group that's at the end of the lysine side chain, is converted into a double bonded oxygen to form an al-lysine, as you can see here. And that al-lysine is actually the molecule that performs the reactions that go into the crosslinks that I've described to you earlier. Now, instead of the lysyl hydroxylase, which was found in the endoplasmic reticulum, lysyl oxidase is found extracellularly, meaning that this reaction occurs only after the collagen has been released from the cell. That release from the cell, of course, requires the hydroxylation reactions that we've talked about before. Now, this reaction that has been produced here allows, as I noted earlier, the aldehyde residues to come into close contact with each other when those chains intertwine with each other. These chains intertwining allows these aldehyde groups to come together and form a chemical bond because they're very, very reactive with each other. They will also react with the hydroxylysines that I showed you on this previous slide. Now, aldehydes of the L-lysine can react, as I've noted before, with the amines of other lysines and form a compound called pyridiniline. Now, pyridiniline is shown on the screen here, and it's a fairly complicated structure, so I'm not going to emphasize that in any important way. But you can see here that there are 3 lysines that have formed this pyridiniline residue that you can see here. This is a covalent structure. And just like if you were to take and braid someone's hair, and for example, didn't put a tie into the hair, what would happen to the braid? Well, the braid will come apart. The braid will not provide the strength needed over time. That's what happens if this cross-link reaction doesn't occur between the 3 strands of the collagen. You put the 3 strands together, you tie it together with a knot at the end, the braid won't come apart. The knot in this case is the pyridiniline. So the cross-linking that you see on this slide is what gives collagen the strength that it needs to do the functions that it has. Now, there are a variety of diseases, as I've noted, that can happen with problems in collagen. One of these is scurvy. Now, scurvy happens as a result of deficiency of vitamin C. Other diseases that happen are a result of genetic diseases or mutations that happen in the coding regions for the various collagens that are there. So some of the genetic diseases involved in collagen synthesis or processing involve osteogenesis imperfecta, which produces brittle bones, arises from deficiencies or problems associated with type 1 collagen. Chondrodysplasia, which is a skeletal disorder, arises from the mutations that happen in collagen type 2. Ehlers-Danlos syndrome, which happens in connective tissues, arises from problems with collagen type 3. And Knobloch syndrome, which occurs affecting collagen in the brain and retina, happens from mutations within collagen type 18. Now, collagen vascular disease can happen also as a result of autoimmune disorders. So one autoimmune disorder here is depicted on the finger of this individual, and these give rise to things like lupus, systemic lupus, and a variety of other things that can cause significant problems for individuals. In this lecture, I've described the biochemistry of collagen, the relationship between its structure and the chemical modifications of collagen that happen, and the requirement of vitamin C to make the overall process occur.